Okay, for defenses and counter defenses, for ectoparasites, you can think of anything on the outside helping to prevent ectoparasites from glomming on. Protective coatings, skin, exoskeletons, etc. And for endoparasites that do gain entry, can be killed by the host's immune system or by biochemical defenses or other defensive symbionts. So we'll look at a few examples of that. The vertebrate immune systems have memory cells which recognize microparasites from previous exposures. And other immune system cells engulf and destroy parasites or mark them with chemicals and target them for later destruction. And so effectively our immune system is designed to recognize invaders, and then based on that, having dealt with them once before, then you've got the memory to deal with them more expediently the next time around. Plants also have defense systems. They have resistance genes, non-specific immune responses such as antimicrobial or antifungal compounds, and chemical signals that warn nearby cells of imminent attack, and chemicals that stimulate deposition of ligand, which creates a barrier for them to spread. This next slide kind of looks at that. So here, let me pause a second because I want to walk through this figure a little bit more here. So here's a leaf with some sort of parasite growing on it. It could be a bacteria, for example. And so that bacteria is starting to attack the cell wall. As the cells damaged by the microparasite infection, they release molecules that stimulate production of antimicrobial compounds. These are phytoalexins. So these are plant-based antimicrobial compounds. Additionally, they trigger development of the of ligand, which is a physical barrier to prevent the micro parasite from spreading. So what they're doing is they're producing a antimicrobial compound, and then they're producing more ligand, and the ligand is a barrier, kind of basically quarantining off that cell, like putting up a thicker barrier so that bacteria can't spread to other members of the cells. And then they also warn the nearby cells of the attack, so they can also produce phytoalexins, or the antimicrobial agents that plants can produce. And then, let me see, microparasite infection. Yep. So it's kind of a three-part response, you might think. So the bacteria is detected, antimicrobial agents are produced within that cell, ligands produced to create more of a barrier between that cell and nearby cells to kind of quarantine off that infection, and then other cells are made aware of that and start to produce phytoalexins as well. So it's a pretty interesting response, just for a plant to be that sophisticated, but obviously, unchecked parasitism is a big issue, and so having defense mechanisms makes sense. And hosts can regulate their biochemistry to deter pesticides. This is sort of an interesting one where vertebrates have a protein called transferrin, which is kind of, it's like transfer and like iron, transferrin. That removes iron from the blood serum and stores it, so bacterial fungal endoparasites can't get to it. So effectively being able to pull iron out of your blood, store it in specialized cells or vacuoles until that bacterial fungal endoparasite's gone, and then re-release it. In some parasites, then it's like this arms race. So then some parasites have developed ways to steal iron from transferrin. And so which could actually be more effective. It's like, I'm trying to think of like an example, but like if you, if something's scared, so it like moves all of its cash into a certain spot, then you just go rob the cash, right? Or store. So store, not meaning like cash, like dollars, or stores in like AM PM, but effectively you're like invoking a mechanism to sequester all of the compound you want in a certain area. Then you can just go and take it out of that certain area. It's kind of interesting. Plants have many chemical weapons called secondary compounds, and we often like refer to secondary compounds. We talk to them in the predator-prey relationships as well, where plants will produce secondary compounds that will make them reduce their chances of herbivory. Tannins are one of them. There's other alkaloids that make them bitter, taste bad, and so there's these secondary compounds that plants can produce. And some animals eat specific plants to treat or to prevent parasite infections. The woolly bear caterpillar will switch from their usual food plants to a poison hemlock when parasitic flies lay in their bodies. I thought that was pretty interesting that caterpillars do that. Chimpanzees, I've heard of that before, so I was less surprised about that, but I was interested that a caterpillar could do it. So chimpanzees will, infected with nematodes, seek out and eat a plant that contains chemicals that kill or paralyze the nematodes. And again, like if you think about, a lot of our modern pharmaceuticals come from plants, and obviously there's a large knowledge base around traditional plants and how they can be used to treat different ailments. Mutualistic symbionts can contribute to defense of parasites. It's kind of a funny one where it's like a parasite fighting off another parasite. So fungal symbionts in some plant leaves protect against pathogen attacks. Evidence is mounting that bacterial symbionts in the human digestive tract can protect against other pathogens. And so it's where then it's in like sometimes these lines get blurred between what's a mutualistic relationship and what's a parasitic relationship. And so if you have a parasite that's stealing resources from you, but it also has some mutualistic benefits, meaning it's also fighting off another parasite. It's kind of funny. It's like that. Would it be the enemy of my enemy is my enemy? Not really. It's more of like the greedy parasite hypothesis. You can think of it that way, where the parasite doesn't want to share its host, and so then it will do a mutualistic function by fighting off other parasites. This is an example from pea aphids with 5 different genotypes represented or exposed to a pathogen fungus, and for each of these genotypes, some aphids were inoculated with a bacterial symbiont, while other aphids lacked the symbiont. Aphids harboring the symbiont survived at higher rates than aphids lacking the symbiont. So here's the red. So these are different genotypes to make sure it's not just specific to one genotype, or it's not we're not measuring the genetic diversity here. We're not measuring a certain allele that one genotype has that other genotypes don't have. So we're including 5 different genotypes. And then the symbiont bacteria is added to the red bars, and the blue bars are ones without the symbiont. And then they're all exposed to a fungus. Survivorship is here, so 100% survivorship is everybody's fine. Even with the symbiotic bacteria, we still have a relatively large mortality rate. And so if we have a survivorship of 50%, we have a mortality rate of 50%. Survivorship of 25%, mortality of 75%. But it's much worse without the bacterial symbiont. That here, when exposed to the fungus, less, that might be 10%, 15%, so less than 10 or 15% survive. Whereas with the symbiont present, survivorship varies, but between 50 and 25-ish%. So it's better with the symbiotic bacteria. Parasite counter defenses. So ectoparasites must penetrate external defenses and toxic compounds produced by plants. challenges similar to those faced by herbivores and predators. Endoparasites must cope with defenses inside the host. Some hosts can encapsulate endoparasites, their eggs, and make them harmless. Some insects have lamellocysts, blood cells that form multicellular capsules around large objects such as nematodes. And so basically they've got specialized blood cells, or their blood cells can encapsulate the entire nematode and sequester it. And parasites under strong selection pressure to come up with counter defenses. And so both of these are actually strong selection defenses. There's a strong selection defense a strong selection pressure rather, to develop defense against a parasite. And there's also a strong selection pressure to defeat that defense or come up with a counter defense. So parasitoid wasps avoid encapsulation by injecting virus-like particles that infect the lamocytes and cause them to self-destruct. Isn't that crazy? So like when a wasp is injecting its larvae into a mite, for example, it's also giving it a virus that then destroys the lamocytes. So it bypasses their defense system while it's giving it an egg that's going to develop into this thing that's going to eat it from the inside out. I mean, this is crazy. Some parasitoids lay eggs covered in filaments that become embedded in the host fat cells where they're not detected by circulating lamellocytes. And so there's less circulation in fat. It's a better place to hide out in. Again, also kind of a crazy one. Not as crazy as giving them a virus that defeats their defense mechanisms. There's I'll try to remember it. There's a book about comparing like animal defenses. It could be beetles. It could be specific to beetles and like war. But this idea of, mounting issuing a virus to disable a defense system is actually, this is an aside, is actually the number one issue in modern defense systems is that we're producing billion-dollar planes that fly on software. You could either produce a billion-dollar plane to combat that or develop software to disable that plane, right? And so these cybersecurity things are the biggest concern right now. Different type of virus, but nonetheless, there's some parallels there. Plasmodium is a protozoan that causes malaria and has a complex life cycle. There's two challenges in the human host. Merozotides multiply in red blood cells, but these cells don't divide or grow and don't import nutrients. Infection by Plasmodium causes red blood cells to have an abnormal shape, and they're destroyed by the spleen. So the Plasmodium, which is a protozoan that causes malaria, has an issue where when it takes over a red blood cell, those cells stop functioning. They're not dividing, and they're not importing nutrients, which the Plasmodium needs, as well as they have an abnormal shape, and so they'll get cleaned out by the spleen. And so they've developed a couple defense mechanisms against that. And the life cycle here is also pretty crazy. So we can start here. Let me get the terms here. So we can start here where it's the sporozyte stage. So we'll start here. We'll pin the mosquito bit. The person, the sporozyte stage, is injected in the human, goes into the liver, where merozytes are formed, and these are released into the bloodstream. They fill into red blood cells. They'll reproduce, burst, infect new red blood cells, and keep going. Ultimately producing gamete-forming cells. I don't have a name for that. but they'll produce a gamete. These are the gamete-forming cells. They don't have a name for that, I guess. But the gamete-forming cells then get sucked up by a mosquito. The gamete-forming cells in the mosquito then produce gametes, male and female gametes, produces zygote that then produces a sporozyte. And then these are released when the mosquito bites the human. These wind up in the saliva of the mosquito. So when the mosquito bites the human, it's then released, and the cycle goes around and around and around. And so this is causing two issues for the Plasmodium, which is once it takes over a red blood cell, that red blood cell stops dividing and bringing in nutrients, and it makes an abnormal shaped red blood cell that the spleen will detect and destroy. So to get around this, Plasmodium has hundreds of genes that modify red blood cells. Some genes cause the transport of proteins to be placed on the red blood cell surface to transport nutrients. So they basically make their own protein to import nutrients into the red blood cell they've taken over. And then others make these knobs that stick to other cells and prevent them from circulating. So they provide these like knobs that will stick to a blood vessel and prevent them from going through the spleen will they'll be cleaned out or killed. And so they're doing two things. One is they're like adding their own machinery to the cell to bring in nutrients, and then they're adding knobs that'll make it stuck somewhere in the blood system so they won't be run through the spleen and detected and killed. Kind of interesting that they're of this development, but pretty interesting. The next section will be on coevolution, so we'll wrap this one up here.